A very warm good evening to one and all. I'm Akulat Sankita today. We welcome you to the Federation of Clinical Pharmacists in India, the fourth presentation on the basic modules in infectious diseases. So I'm introducing you all on behalf of our FCPI team members. So today's topic is on microbiology and antimicrobial resistance, focusing on the non-fermenters, especially Pseudomonas aeruginosa and Acinetobacter species. So before moving on to the session, I would like to inform you that the certificates will be provided within 7 days after the end of the session. So without much time, let's move to the session. So to introduce, I'm Sangeeta. I'm professionally a clinical pharmacist and have more than 5 years of clinical and research experience. So thank you all for joining. And so we have already discussed the gram-negative bacteria, the basic microbiology and antimicrobial resistance of gram-negative bacteria, focusing on Enterobacteriaceae in the first session. And today we are focusing on the non-fermenters, especially the Pseudomonas and Acinetobacter species. So we will be discussing on the classification, characteristics, differentiation, the focus of infection, and some of the risk factors, and some short insights onto the basics of resistance mechanisms. So basically, why we focused on Pseudomonas and Acinetobacter is mainly because these are considered to be one among the escape pathogens. So escape pathogens includes escape. The term escape is actually an acronym. E stands for Enterococcus, S for Staphylococcus, K for Klebsiella, A Acinetobacter, P Pseudomonas, and E Enterobacter. Actually, these organisms are considered to be difficult to treat. These are highly resistant strains, and they are considered to be one of the difficult to treat organisms, and it's causing a lot of mortality and morbidity, and hence this needs to be dealt with alarmingly. As you can see, the Aceneto and Pseudomonas aeruginosa, the mortality among these two organisms are significant, and it needs to be considered important. And according to the Center of Disease Control data, the prevalence of these organisms, the estimated case and its associated deaths are very high. It's around 2,700 in 2017 versus 700 for Acinetobacter in 2017. So as per the global antimicrobial surveillance data, the CRAB, or the carbapenem resistance Acinetobacter strains, are estimated to be 60%, and it is very high in low middle income countries. And this we have already discussed in the previous session. So this has already been discussed. As you know, just to brush up your knowledge, whenever a patient presents with a sign or symptom, first thing what we do is to check if it's an infection or in non-infectious etiology. Once we conclude that this is an infection, first thing we do is microbiological sampling. So what we do first is gram staining, just to understand if it's a gram-positive or a gram-negative bacteria. Once we identify this is a gram-negative bacteria, the next thing we will be checking whether this is a lactose fermenter or a non-lactose fermenter. So here in this case, both the Acinetobacter and Pseudomonas are lactose non-fermenters. They do not ferment carbohydrates, lactose, nothing. And another feature which makes them unique, another feature which differentiates between them is the oxidase test, whether it is oxidase negative or oxidase positive. So we have already discussed what is oxidase testis in the previous session. So hope you know, like it is the ability of an organism to produce oxidase, cytochrome oxidase enzyme. And there are specific tests to identify if that particular organism is negative or positive. Here in this case, the Pseudomonas has the ability to produce cytochrome oxidase enzyme, and hence the test will be positive, whereas Acinetobacter will be negative. So let's discuss more about the Pseudomonas species. So this is basically the microbiology part. So one thing which actually makes them unique is they have a particular, there are certain factors which makes them unique, like they have the order, the typical order these produce, like it has got a sweet or grape-like or a corn taco-like order. It's basically because of its production of aminoacetophenone. Another important feature is that they can produce certain pigments like pyocyanin, pyoverdin, pyruvin, pymelanin, which actually gives them a typical color, which is greenish fluorescent color in the culture. Apart from that, these are oxidase positive, as I've already mentioned. They are catalyst positive. And another important feature is that these grows well at 37 to 42 degrees Celsius. So 4 major things are: 1, it has got a typical order, sweet grape-like, corn-like taco order. And then it has a particular color, the fluorescent greenish color due to the pigments. It has the ability to grow at 42 degrees Celsius, and they are oxidase positive. Apart from that, there are various other features as well. So although this is aerobic, they can actually grow in an aerobic condition in the presence of nitrate. They have got a pili or a fimbriae, which helps them to attach to the host surface. Now, in certain cases, when the patient has cystic fibrosis, the culture usually appears as mucoid colonies. And this is basically because of the production of alginate. Apart from that, there are certain things like lipopolysaccharides, exotoxin A, exoenzyme STUY. All these are responsible for its endotoxic property, and certain things causes tissue necrosis. And that is why in certain cases it says that when Pseudomonas causes eye infection, it actually has the ability to cause irreversible damage and vision loss, mainly because of this necrotic feature. Now, this is very important. For both Pseudomonas and Acinetobacter species, they have an ability for quorum sensing and biofilm formation. Quorum sensing means these organisms can release certain things like acylated homoserine lactones, and this diffuses into the cells of this bacteria. The same organism produces these things and diffuses into the cells of the bacteria, and then it actually triggers or promotes certain translational regulators within them, and thereby develop biofilm formation. So these biofilm formation is actually a protective barrier for this organism against antibiotics. These biofilm will be formed, for example, in certain cases like when patient uses a medical device, for example, ET tubes, biofilm is produced, and these colonies will survive within this biofilm. It's very difficult for the antibiotic to penetrate through this biofilm and kill the bacteria. So this is one of their surviving mechanisms. So let me tell you, biofilm is not just for Pseudomonas, it's for Acinetobacter as well. So Pseudomonas, this is the gram stain of, this is how it looks. These are rods, gram-negative rods, and this is how it looks. And these are its cultures. As you can see, the greenish fluorescent color, they grow well at 42 degrees Celsius. They have a corn-like, taco-like order, and this is the positive oxidase test. And you can see the mucoid colonies could be because of the alginate production. So these are yet another examples: green-gray color colonies on blood agar plate, production of pyocyanin pigments. And now moving on to the Acinetobacter. So as you know, Acinetobacter has been causing a lot of mortality and morbidity. You may have seen within your hospital itself. The mortality rate is estimated to be more than 70%. Although they are rod-shaped, they appear as cocobacilli. Now, they grow well in most of the medias. And they have a similar resemblance to the Neisseria in cultures, but how we differentiate is basically because based on this oxidase test. So Acinetobacter, unlike Pseudomonas, it is oxidase negative, whereas Neisseria is oxidase positive. And Pseudomonas is also oxidase positive. So just like Pseudomonas, these originates in moist medium. These originates in the water of room humidifiers, vaporizers, and therefore infection prevention control practices play a very crucial role in containing the containment of this organisms. So these are some of the tests we can use to differentiate the Acinetobacter species. Like all these tests will be positive if it is Acinetobacter, whereas all these tests will be negative. Now, this is just an example. So you can see Pseudomonas was rod-shaped, whereas this one is it has a cocobacillary appearance. And these are some of the smooth round colonies in McConkey agar. Now, these are some of the differences. So Pseudomonas is motile, whereas Acinetobacter is non-motile. Pseudomonas is rod-shaped, whereas this one resembling to cocobacilli. Now, this has got a typical order, and some literature say this has a fish-like order. Both of them appears to be in pairs or short chains. Pseudomonas have a typical pigment which gives them a color. They both lives in moist environment. They do not ferment carbohydrates, and therefore they are non-fermenters. They are catalase positive, both of them. One is oxidase positive, another one is negative. Both of them can cause biofilm formation, and both of them has a pili which can attach to the host surface. Now let's discuss a few questions. So a 17-year-old girl with cystic fibrosis has slight increase in her frequent cough and production of mucoid sputum. A sputum specimen is obtained and plated on routine culture media. The predominant growths are GNB that form very mucoid colonies after 48 hours of incubation. These bacilli are oxidase positive, grow at 42 degrees Celsius, and have a grape-like odor. So what could be the organism be? So let us know by typing in. So we'll give you some time. What could be the organism? So this is gram-negative bacilli. They have a mucoid colony. It could be because of the production of alginate. They are oxidase positive. They grow at 42 degrees Celsius, and they have that grape-like odor. And that is obviously Pseudomonas aeruginosa. Next question. A wounded civilian undergone amputation of both her legs below the knee and was placed a suprapubic tube to repair damage to her bladder. She presented with recurrent UTI that has not responded to conventional antibiotic regimens for community-acquired cystitis. Gram stain of the urine showed gram-negative cocobacilli. When cultured, this organism does not ferment carbohydrates, so obviously non-fermenter, does not hydrolyze urea, does not reduce nitrates, and does not make hydrogen sulfide. The organism most likely could be, please type in your answers. So this is gram-negative Cucubacilli, and therefore, obviously, Acinetobacter baumannii. Okay, so let's discuss about these organisms a little bit more. So both of them, this we have already discussed previously, then which are the normal bacterial flora. As you can see, both of these, Acinetobacter and Pseudomonas, both of these are actually opportunistic pathogens. They are not commonly seen as a commensal, especially for Pseudomonas. Except for Pseudomonas in certain cases, like in upper respiratory tract in the upper respiratory tract and GI tract, where they are colonizers in certain cases, but not Acinetobacter. But both of these are usually opportunistic pathogens when there is an abnormal host immune response. So both of them have the ability to generate in a moist medium. So whenever the patient is immunosuppressed, for example, when they're taking an immunosuppressant or after chemotherapy, or patient undergone a surgery, after prolonged hospitalization, taking off antibiotic exposure, or a medical device in situ, like a ventilator, suprapubic catheter, or there is a breach in infection control, prevention control practices. These generate in a moist medium and has the ability to invade. These colonizers will become pathogenic and especially causes nosocomial infections, both of them. So they usually appear in contaminated water, respiratory equipments, and it could be the cleaning solution, sinks and mops could be one of the transmitters. And these are some of the infections that it can cause. So let's discuss some of the infections and its risk factors. So the dark blue color is the infection that these organisms cause, and these light blue color are the causes for it, some causes or risk factors for it. So, for example, Pseudomonas aeruginosa can cause cystic fibrosis, and this can hematogeneously spread to other sites. And one other cause for the respiratory tract infection could be invasive ventilation. As we already discussed, when the ventilator, the ET tube is in situ, these organisms will, and when there is a breach in IPC practice or whatever it be, these organisms can invade the host and can colonize within the medical device, form a biofilm, become a pathogen, and can cause the infection. Now, colonization. So Pseudomonas, as you know, it is colonizer in GI tract, and when the patient becomes immunocompromised, it can cause necrotizing enterocolitis. Now, another causes could be catheterization, instrumentation like urinary stent, other urologic procedures. All this can cause UTI. Now, again, the medical devices like intravenous catheters, and again, when the patient becomes immunocompromised, it can cause, the hematogenous spread can lead to the bloodstream infection, sepsis, multi-organ dysfunction. And another thing is intravenous drug use. In certain cases, intravenous drug use has led to infective endocarditis. Extension of parameningeal structure, diagnostic procedures, surgical procedures, and hematogenous spread from other primary sites can cause meningitis and abscess, even in infants. Now, again, hematogenous spread, surgery, overlying soft tissue infections, and all of these are some other reasons for the skin-borne and joint infections. Now, as you know, both these organisms generate in a moist medium, and especially Pseudomonas aeruginosa, they can be one of the these hot tubs, whirlpools, swimming pools, and other type of common baths could be a hub for these organisms. And in certain cases, it can cause dermatologic infections like hot tub rash with dermatitis or folliculitis. Now, trauma prolonged use of contact lens, exposure to ICU environments, or another other factors could be one of the reasons for eye and ear infections causing bacterial keratitis, scleral abscess, endophthalmitis, etc. So this was all about some of the sites, the infections caused by this bacteria and its the risk factors. And now we will be discussing the basics of resistance mechanisms. I already told you these infections are difficult to treat, especially because they are highly virulent and they easily become resistant. The emerging resistance is one of the risk factors which make them difficult to treat and is leading to high morbidity and mortality. So these are some of the resistance mechanism by which these becomes resistant strains. So one reason is loss of porins. You may know the porins are channels through which the antibiotics can enter into the organism. So when there is a loss of these porins, the antibiotics cannot penetrate through and the bacteria can survive. So for example, beta-lactams and fluoroquinolone is facing this issue for Pseudomonas and Acinetobacter for Acinetobacter. Another thing is efflux pump system. So upregulation of efflux pump system in both these organisms will throw out the antibiotic and will not be able to kill the organism. And for example, is TG-cycline and imipenem resistance in Acinetobacter species. Third one is enzyme mediated. So the production of class C AMC beta lactamases, which is inherent. So that is why imipenem first and second generation cephalosporins are resistant, will not be used for Pseudomonas species. And acquired beta lactamases, acquired could be because of many reasons like previous antibiotic exposure or subtherapeutic concentration, and you know what could be the reasons. So that is one of the reasons for developing these kind of enzymes, class A, B, C, D, so which we will be discussed in the coming sessions, which I'll discuss with you later. So OXA 48 strains is one of the example. Aminoglycoside modifying enzyme development for both these organisms. Now, target site mutations. So alteration in the target sites like penicillin binding protein in Pseudomonas causing fluoroquinolone resistance, or point mutation in the DNA gyrase for quinolone resistance are some examples. Now, last but not the least, quorum sensing and biofilm formation, which we have already discussed, is yet another reason to say that these organisms are developing resistance and are difficult to treat. So this was all about the basics of non-fermenters for a very short session. So we discussed its basic microbiology, the risk factors, and the short resistance mechanism, but definitely we will be discussing onto the antimicrobial resistance in detail and its management, the strategies based on antimicrobial stewardship approach in the upcoming sessions. So these were the references we have used, and kindly let us know if you have any questions, or kindly reach us with your questions in this email. And before that, let me tell you that the feedback form will be available at our website after the end, towards the end of the session. So now it's open for questions. Thank you. So we have a question, a relevant question. So to differentiate between enzyme-mediated resistance of Pseudomonas aeruginosa and Acinetobacter. So So as I already mentioned, so some enzymes are inherent and some enzymes are acquired. So inherent enzymes like the class C AMC beta lactamases. So these are inherent, and it can so the examples like imipenem, first generation, second generation cephalosporins are inherently resistant, so it is not beneficial for Pseudomonas treatment. And certain acquired beta lactamases like class A, B, C, D enzymes, there are a lot, not just OXA48, there are a lot of other strains as well, which are acquired, and therefore their management will be different. So in this session, we are just discussing the basics, and in the upcoming sessions, we will be discussing this in more detail. So similarly, aminoglycoside modifying enzymes, so like N-acetyl transferases, O-nucleotidyl transferases, or phosphotransferases, are also contributing to resistance among these organisms. So that was all about that, and let me know if you have any other questions. We are also getting other questions like what are the treatment and management. So that's not the scope, that's not coming under the scope of this session. So we'll be discussing the management in the upcoming sessions, and you can definitely enroll to that. And yeah, another question is, we discussed about biofilm formation. So which are few antimicrobial agents that destroy biofilm easily. So it's not about destroying biofilm easily. There are certain antibiotics which could be penetrating through the biofilm. So colistin is obviously one among it. And so that was all about our questions. So we'll wait for a few more questions. What are the major risk factors of increased mortality rate of these organisms? So, as I already mentioned, the risk factor is basically because of its emerging resistance. One is basically breach of infection prevention and control practices. Second thing is inappropriate management, prolonged exposure to antibiotic inappropriately, or prolonged hospital stay, prolonged prolonged placement of the medical devices could be another risk factor. So all of these are majorly contributing to the high morbidity and mortality. I think that was all. We have got the questions. So with that, thank you so much, and kindly reach us if you have more questions in this mail ID. And as I already mentioned, the feedback form will be available at our website and by the end of the session. And thank you so much.